Well, thank you all for being here for the first panel session of this semester's Honors Thesis Symposium. We're super excited about the lineup for today. And our first panel is titled, There's Something in the Water. So we did have 2 water presentations this semester, so they are both here to share with you their findings this morning. And I will let them introduce themselves. Deanna and Dominic are both going to tell you a little bit about who they are, their majors, their interest in the topic, and then they're going to share with you what they found as part of their thesis project. I always forget to say who I am, and I did it again. I'm Megan Hughes-Darzo. I'm the director of the honors program. So thank you for being here. I see a lot of familiar faces and some I don't know. And also, I also forget to tell people that there is tea and coffee in the next room, but I remembered this year. So if you want some, feel free to sneak out and get some tea and coffee or water. And we'll go ahead and get started, all right?
Good morning, everybody. My name is Dominic Baca, and I'm a senior at Metro, and I'm majoring in environmental science with a concentration in hydrologic science and water management. And this morning, I'll be telling you about my thesis research. And I was actually fortunate enough to do my thesis research in Costa Rica as a part of a research experience for undergrads through the Organization of Tropical Studies. While I was in Costa Rica, I was a part of the streams team, and I was doing aquatic ecology research, and my specific area of focus was microbial respiration on woody debris across a natural phosphorus gradient and experimental pH gradient. And we'll just jump right in. So first, I'd like to give you a little bit of context about the area and the region in which I did my research. So I was at La Selva Biological Station in the northeastern region of Costa Rica. And La Selva Biological Station is a nature conservancy. It's roughly 1,500 hectares of protected land, and the climate there is Neotropical rainforest. And La Selva is recognized globally for its rich biodiversity and also as one of the most productive field stations for tropical forest ecology research. And as you can see on the map shown by the star, that is where La Selva is located in the country of Costa Rica. And this scenic bridge behind me is a very iconic photo taken from the biological station, and that's pretty much what it's known for, is their massive suspension bridge. So water quality at La Selva is a lot like water quality everywhere else around the world in that it is highly variable due to lots of different factors, whether they be environmental, biological, or chemical factors. So there are many water quality parameters that I could have assessed, but the water quality parameters I chose to look at and investigate in my research were the volcanically active landscape of the region, and the volcanically active landscape created geothermally modified groundwater, and then that groundwater would then seep in to the streams and affect it differently because it has a different chemical makeup. I also looked at nutrient availability, specifically phosphorus availability, as well as pH and different detrital interactions within the water systems, specifically looking at coarse woody debris and its effect on the biological oxygen demand and the decomposition of woody debris in relation to the water quality dynamics. And this is a photo that's more of a river in Costa Rica. The streams that I studied were much smaller than that, and that is an actual photo that I took. And a lot of these photos were photos that I took, so that's pretty cool. So as I mentioned, there are 2 different types of groundwater at La Selva. There is geothermally modified groundwater, and there's also just kind of regular water, that's regular groundwater, that doesn't really have anything special about it. In this image on your left depicts how that groundwater is distributed spatially. So there's kind of a 50-meter rule in the distribution of groundwater. So above 50 meters in elevation, the streams typically do not receive inputs of geothermally modified groundwater. And these streams are characterized by low pH, a low buffer capacity, and lower nutrient concentrations. And for my purposes, this meant low phosphorus concentration. Whereas streams that were below 50 meters in elevation above sea level, these streams did receive inputs of geothermally modified groundwater. And these streams were characterized by a relatively higher pH, a better buffer capacity, as well as higher nutrient concentration, so higher phosphorus. And in this image, you can see that La Selva is right here. And so there is geothermally modified groundwater, and there's also local groundwater. And the geothermally modified groundwater has higher nutrient concentrations, such as chlorine, silicone, and also phosphorus, which was my study nutrient. Another parameter that I looked at is coarse woody debris and the importance of coarse woody debris. So coarse woody debris in general is just wood in an aquatic stream. And it's really important because it acts as a primary source of carbon. And that carbon drives aquatic food webs, and it can affect aquatic ecosystem dynamics. Therefore, the availability of carbon and the availability of coarse woody debris has a great impact on aquatic ecosystem dynamics. And as you can see in these photos, also that I took, the region, La Selva, is very heavily forested, and there is lots of wood shrapnel around because the area is highly subject to tree fall events, whether they're natural tree fall events or whether they're tree fall events that are induced by severe weather events. There was actually a really, really large wind, almost tornado-like event that occurred the week before I traveled to Costa Rica, and that is the cause of all of this woody debris that you can see in these massive tree falls. So the wood in and out of the streams is very important as it affects the entire food web dynamics. So next we'll talk about microbial respiration. So microbial respiration is simply microbes in the water taking in dissolved oxygen and producing dissolved carbon dioxide. So it's basically just microorganisms breathing in the water. And a good way to quantify this is through a biological oxygen demand test. And these tests typically have time periods prescribed to them. So for my study, I had a 5-day and a 10-day biological oxygen demand test. And the way you calculate biological oxygen demand is by subtracting a final dissolved oxygen measurement from a final dissolved oxygen measurement. And the biological oxygen demand test is really important because it tells you how much oxygen is needed in a system in order to keep it aerobic and in order to keep the organisms within the water that do breathe alive. And if that oxygen demand is not met, then the system runs the risk of going septic, in which case aerobic respiration cannot be supported. And this image just shows a microbe taking in dissolved oxygen and producing dissolved carbon dioxide, which is microbial respiration. So after all of these factors were taken into account and considered, I landed on my hypothesis in my research question. And that is the interaction between phosphorus and pH will have a statistically significant impact on the microbial respiration and decomposition of coarse woody debris. So modern aquatic ecology tells us that the more nutrient-enriched an environment is, the more microbial activities can occur, such as respiration or decomposition. And modern research also tells us that more microbial activities can occur when a water body is at a more neutral pH, a pH around 7. However, there's no research that evaluates whether or not there's a significant interaction effect between nutrients and pH. So that's really what my study was aimed at, to evaluate if there is an interaction effect between pH and phosphorus on microbial respiration and decomposition of coarse woody debris. So next, so in order to evaluate my research question and my hypothesis, I needed two things. First, I needed a phosphorus gradient, and second, I also needed a pH gradient. So thanks to the naturally occurring inputs or lack thereof of the geothermally modified groundwater, nature provided me with its own phosphorus gradient because the different streams at La Selva have different phosphorus concentrations naturally due to the GMG. And excuse me. And so I studied 3 different streams, and each different stream had a different phosphorus concentration and represented a high phosphorus stream, a low phosphorus stream, and a medium phosphorus stream. And although each stream had a different pH naturally, I decided to modify the pH within each sample set with pH treatments in order to see what was going on at different pH levels for each individual stream. So for pH adjustments, I had an acidified treatment, which involved adding an acid to my sample, which lowered the pH by 1 log unit. I had a buffer treatment, which involved adding a base to my samples, which raised the pH by 1 log unit. And then there was a no treatment, so I also assessed the natural pH. And these photos are photos of my actual study sites. This stream is the low phosphorus stream, and this stream is the medium phosphorus stream. And I didn't take a photo of the high phosphorus stream. So in order to quantify decomposition of my wood samples, I used a percent mass remaining equation. And this simply involved massing the wood samples before the experiment and then massing them again after the experiment. And then plugging the initial and the final masses into the percent mass remaining equation that's shown on the screen. And that gave me a percentage of how much mass in the wood sample was remaining after the experiment had taken place, which also you can deduce how much mass was decomposed throughout the experiment. And again, that is the actual photo of the actual site where I collected my wood samples. And I used a Pentaclethra macrolobus tree species, which is common in the region, and it had just fallen, so it was fresh for the pickings. So after everything was considered and evaluated, I was ready to start my experiment and design my experiment. So as I said, I studied 3 different streams, and I assayed each stream individually. And a stream assay involved going to the stream, filling up my 18-liter carboy, strapping it to my back, and then hauling it back to the laboratory. In the laboratory, I would fill up my BOD chamber bottles, and then I would perform pH adjustments, I would add the woody debris, and along the whole time, I was recording data, such as dissolved oxygen, pH, temperature, and etc. And in order to give you a better understanding of exactly how I designed my experiment and what I did, I've created this graphic. So each blue circle represents a biological oxygen demand sample bottle, which is shown right here. And they are glass and they are stoppered at the top so that no gas exchange can occur within the bottle. So I had 22 sample bottles, and the first step was to fill each bottle with water from the stream. The next step was to divide the bottles into the different time period of tests that I would perform. So I had a 5-day time period test, and I also had a 10-day time period test. After they were divided and filled, then I did the pH treatments. So the pink dots represent the acid treatment, which lowered the pH by 1 log unit. The purple dots raised the sample pH by 1 log unit, and then the blue dots had no pH treatment, whereas the orange dots acted as my control, so they received no pH treatment, and they also received no woody debris sample. And the final step was to add my woody debris sample to the chamber bottle, and then to incubate the bottles in a cooler for their prescribed amount of time, whether it's 5 days or 10 days. And then after that, I collected all my data and I was ready to perform my statistical analysis. So I used a one-way ANOVA to test each individual variable, so pH and phosphorus, and then I used a two-way ANOVA to analyze if there was an interaction effect between pH and phosphorus. And an ANOVA test is just a statistical analysis test which analyzes a data set in order to see if there's a significant variance in your results or not. So shown on the board on the left is the pH treatment ANOVA, and my results, let me explain the colors. So the red highlighted colors indicate that the results were not statistically significant, and anything highlighted in green indicates that the results that I received were statistically significant. So on the left, and this is for my 5-day biological oxygen demand test. On the left, we have the pH treatment one-way ANOVA, and my results were not significant. And on the right, we have the phosphorus concentration versus biological oxygen demand one-way ANOVA, where my results were statistically significant. And in order to evaluate the interaction between the two, my results were not significant, indicated by the not significant p-value. So that was for my 5-day biological oxygen demand, and my next test was the 10-day biological oxygen demand, where again I received results that indicated that pH was not significant, phosphorus was significant. However, with my 10-day test, I did receive results that the interaction between phosphorus and pH was significant. So that's really cool because that's what I was looking for. That was my research question and my hypothesis. So I did get one significant value. And that was for my biological oxygen demand tests. And now I'll move on to my decomposition tests, my 5-day decomposition tests. Like the biological oxygen demand test, I showed that pH was not significant, where phosphorus was significant. And again, there was no significant interaction between the two in regards to wood decomposition for the 5-day test. And in the 10-day test, I got the exact same results, where pH was not significant, phosphorus was significant, and there was no significant interaction. So that was kind of a lot to throw at you. So here's a nice table of what I just said. So here we have the different tests that I performed, the BOD 5-day, 10-day, and the decomposition 5-day and 10-day. Here are the pH results, where pH was never statistically significant in any of my tests. The next column is the phosphorus results, where phosphorus was statistically significant in its impact on microbial respiration and decomposition in every test that I did. And lastly, my big research question, the interaction between the two. And the results indicate that only the biological oxygen demand 10-day test yielded statistically significant results, where all the other tests did not yield statistically significant results. So why is all of this important? As I said at the beginning, water quality everywhere and at La Selva is highly variable due to different factors, whether it be geothermally modified groundwater, carbon dioxide, water treatment plants, or anything above. It's really important to understand the interaction between water quality and ecosystem dynamics, because we would like to keep all of our ecosystems as happy, healthy, and productive as we can. And that's why it's important to study these things. It's also important to note that this was just one small piece of the puzzle. I looked at phosphorus and I looked at pH, but there's a plethora of different factors that I could have assessed. And that leads me to my future inquiries. So after doing this study and having some research experience under my belt, I thought of different things that I could do beyond the study and to further this study. So first of all, my experiment was not perfect and it did involve some experimental error. So something I would like to do in the future would potentially be to replicate this experiment specifically for the high phosphorus strain, where I got some kind of wonky results in the decomposition portion of my study. And I would also like to explore longer biological oxygen demand test trials. Since I did get a statistically significant result in the 10-day trial, that could insinuate that there is actually an interaction going on between the phosphorus and the pH. However, that interaction could take a long time to take place, which might be the reason why I got that result in the 10-day test and not the 5-day test. So it could be interesting to explore longer time periods. And there's also lots of different factors that I could include in this to actually see what's going on in the water chemistry. And that could look like something like looking at different nutrients such as nitrogen, or also looking at any sort of heavy metals that might be in the water. I'd like to give acknowledgements to my field advisors in Costa Rica, Nicholas Marzoff and Alonso Ramirez, who are critical in helping me through this process. And I'd also like to give recognition to my home advisors, Dr. Sarah Schliemann and Dr. Randi Brazeau, for all of their help through the process. And also to all of my sources of funding for this research, and that is from the Organization of Tropical Studies and also the National Science Foundation. I can now take any questions.
Wow.